Yeah. Who can do the process of it. They can't. It's important thing to them.
But I don't want them to call out in the middle of your talk.
Okay. Okay. Good to see it. Yeah. Yeah. Call me down here. Jesus. So he called me down and then he said, and then okay,
Okay, good afternoon, everybody. Zoomers, you are muted. Please stay muted. Put your questions in the chat, and we will get to your questions at the end. If you're in the room and you have a cell phone, if you can please silence it, would be greatly appreciated. And Dr. Kinrad is here to introduce our speaker.
Thanks, Pam, and thanks, Pushpa and people at ILR for setting all this up. I am really happy, thrilled to introduce Dr. Bert Flanegan. Bert has been a colleague at UF for, I think we were estimating about 45 years. It's probably actually maybe longer, but long time and great person to work with. I've run experiments by him on several occasions and really had great advice. So Dr. Flanegan got his undergraduate degree at Florida State, where he also got a master's degree. He then went on to get his PhD in biological chemistry at the University of Michigan. And this was followed by a postdoctoral fellowship at MIT in the lab of none other than the Nobel Prize winning David Baltimore. So he got superb training and credentials. He then joined the faculty here at UF 77 or 78, somewhere.
78.
78. So just about the same time I did. And had I would say, exponentially rising career with a high number of NIH and NSF grant fundings and excellent publications to become a full professor probably in less than 10 years, which is fast. And then chairman of the Department of Biochemistry for some was it 24 years?
24.
24 years. Oh, absolutely sparkling, spectacular career. And his passion is poliovirus, which is sort of interesting that one is passionate about a disease that you're trying to eliminate. But he's made a major contribution along those lines, and I'm going to turn everything over to him to tell you about it. Thank you.
Well, thanks, Ken. I appreciate that. Basically, I've been interested in RNA viruses, and at the time I was a postdoc, polio had been and remained the primary, model system for studying, RNA viruses and RNA virus replication. So it's been, and it's a type of thing that was a model system, and there was a lot known about how it worked. And so there were a lot of, people working in the field and a lot of information to build off that base. So it ended up being a good system to study. I've studied other viruses like that are human enteroviruses like Coxsackie virus. Spent some time studying hepatitis C virus, which is a little different family. But basically staying in that general area. So today I want to talk about the poliovirus vaccines that are required and have been used and have been spectacular really in terms of controlling poliovirus and have reduced it to a very small number of cases, but it has not been eradicated, and so it still remains an issue. And even though those vaccines have worked very well, they do have some shortcomings. They were developed really in the 1950s and early 60s, and so the science that was available in terms of developing those viruses. When we'll talk about those vaccines, a lot of advances have been made, and we're trying to develop better ones. So basically, just to give you a thumbnail sketch, I have to teach you a little bit about the virus and how it works. And so basically, it's like I said, an RNA virus. This is a representation of the virus from studies with X-ray crystallography. And the virus has an RNA genome. It's just a single piece of RNA and in the genome, and it's basically like a piece of what's known as messenger RNA. It can be translated and copied directly into proteins. This virus has no, it's just a protein shell packed, that's around the RNA genome and has no membrane envelope. So a lot of viruses like influenza and others have a membrane envelope, which helps them in attaching to cells, but it also makes them less stable in the environment. So these coronaviruses are very stable in the environment. And for case of polio, humans are the only natural host. And we'll talk about that a little bit more because that makes them a candidate, at least for trying to eradicate the virus. The other thing that's important to know about this type of virus is that there's only a small fraction of the infections, about 1%, that ever result in paralytic disease. So actually most of the infections are silent infections and you actually don't have any symptoms at all. About 10% of the people who get infected have mild symptoms that would be characteristic of a viral infection, some aches and pains and a little fever perhaps, but nothing in terms of paralysis. So these viruses, even if you don't have any symptoms, you still shed the virus, which you can spread to others. Virus spreads from person to person by what is called the fecal-oral route. The virus is in high concentration in fecal material, and the virus is stable, and so it really depends on the spread. If you have poor public sanitation and poor personal sanitation, then you could, it easily spreads. In fact, it spreads pretty efficiently in areas where public sanitation is poor, as I make in the last point. Okay. So what about the vaccines? So there are two vaccines that have been used for a long time. And the original vaccine was created by Jonas Salk. And so it's an inactivated vaccine. So basically, they grow up, to make the vaccine, you actually grow up a lot of virus, and then you chemically inactivate it. So the chemical inactivation leaves the particle, the virus particle, intact, but it modifies the components of the virus, and basically it is no longer able to replicate. But it serves as a good antigen for the body to carry out an immune response and make antibodies to the virus particles. But it's no longer infectious. So basically, one of the biggest advantages is it's safe, since there is no infectious virus present in the vaccine. It provides excellent systemic immunity, so you have antibodies in your blood, and can be used in immunodeficient and immunosuppressed individuals, because basically you're just, the virus doesn't replicate itself, it's just generating the immune response. Now, disadvantages are that it requires an injection, so it's given by a shot, so it requires someone with some skill in terms of being able to give the shot, and then you need the needles or the other devices that you can use to inoculate someone with the vaccine. It's relatively expensive compared to the second vaccine, and there's a second vaccine which is called OPV, or oral polio vaccine, and so the IPV, this vaccine, is about 5 times more expensive. And when you're trying to vaccinate everybody in the world, you're talking about hundreds of millions of people to vaccinate. So a little bit of difference in cost is a big difference in terms of the total cost of using the vaccine. This particular vaccine, although it's very safe, is less effective in preventing the spread of the virus because of reduced immunity in your intestinal tract. So basically, you can be vaccinated and just have to think about, how does this work? When you get infected with polio, you swallow the virus, it goes into your intestinal tract, and then it replicates in the intestinal tract. But in a lot of cases, it never gets into your blood. And so to cause paralysis, it has to get into your blood and then circulate and get to your central nervous system. And if that happens, then you can have your motor neurons get infected, and then they get killed, basically. And so that's where you get the paralysis. But if it just replicates in the intestinal tract, it really doesn't cause any paralysis. But you can shed the virus, so you can get infected. So people who don't have any clinical symptoms, they're still shedding the virus. But you don't produce, since the virus isn't really, in other words, it doesn't, in the case of IPV, that virus you actually take orally, so it actually replicates in the intestinal tract, and you get local immunity. Whereas IPV, you get a shot in the arm, and the virus doesn't ever replicate, so you don't develop any local immunity in the intestinal tract. So the other vaccine, which I'm going to talk about next, basically gives you a longer protective immunity in the intestinal tract. Now, there is a hazard associated with the current IPV vaccine. And so if you're making this vaccine, you have to make it in a large production facility. So you're growing up huge quantities of the wild type virus. And then in that same facility, you inactivate it. It's a fairly complicated, long process, and you have to do it just right. You have to make sure you kill all the virus, but without destroying the capsids. And so the virus can escape from those facilities. And if you are in a world where you're trying to get rid of polio from spreading, any release from the production facility is a hazard. Sometimes, Nothing's perfect, and the production facilities are not perfect. So sometimes they accidentally release something that's contaminated with virus. And so that's going to be a hazard if in a world where we're trying to get rid of all of a particular virus. Okay, the other vaccine is called OPV. This is the oral vaccine, and I've already referred to this some. As I just said, it provides strong systemic and intestinal immunity. It's inexpensive, approximately, it's pennies, basically 20 cents per dose in the reference I looked up. You can administer it orally, so it's just it's a little, dropper, and you just put some drops in the person's mouth. So basically anybody can deliver it. The virus is a live virus, and so it replicates in the intestinal tract, and you get some secondary spread. That's both good and bad, as you'll see. The good part is you vaccinate more people than you actually administer the vaccine to, so you get some wider spread, which is basically what's called herd immunity. The more people in a group that are vaccinated and protected, the harder it is for the virus to spread in that group. And so that protects the whole population as long as you get the herd immunity high enough. So it's relatively safe and very effective in controlling outbreaks of wild-type virus causing poliomyelitis. The disadvantages are the reversion of the attenuating mutations that cause rare cases of vaccine-associated paralytic poliomyelitis. And you can see, I'll show you in a little bit, that's one of the biggest problems right now that we're dealing with this vaccine. Remember, this was made back in the 1950s and early 60s. We didn't have any sequences for the RNA. Basically, mutations were picked sort of randomly that made the virus less virulent. And for a long time, we didn't even know where those mutations were at or what part of the function, was being inhibited during replication. So basically, those mutations can go back to their original form as the virus replicates by the process called reversion. And so that can cause problems. And as a result, we get what's labeled here as cVDPV, and that's circulating vaccine-derived poliovirus. That's actually our biggest problem right now in using this oral vaccine, because that means the vaccine has derived, it's now pathogenic, it has mutated and reverted, so it's now pathogenic, but it's also acquired its ability to circulate in the population very efficiently. So it's basically not much different than wild-type virus when this happens. And so this is in particular happening in areas with low immunity against polio and in places where there's poor sanitation. So you got if you have a population where there's not much immunity because people haven't been vaccinated or previously infected, and you have poor sanitation, so the virus actually spreads rapidly person to person in that environment. And as it spreads and replicates, it can mutate, and so it regains the ability to spread, replicate, and also cause paralysis. So, and in this case, you definitely don't want to use this. It's a live virus vaccine. You don't want to use it on immunodeficient or immunosuppressed individuals. And this, even in the production of this particular vaccine, the testing for safety testing is done in monkeys. So you're, try to avoid if, at all possible. Okay, so I just want to tell you, this is not going to be like my virology class at UF, but I want to tell you just enough to get the idea of how this works, how the vaccine works. Okay, so this is a cell, a hypothetical cell, which is being infected with poliovirus. Okay, so for the virus to replicate, it has to get inside of the cell. And for it to do that, at step one, shown here, the virus needs to attach to a specific protein that's on the surface of the cell. So this is a viral receptor. This is only in human cells. So human cells are the only cell type that will support viral replication. Primates, there are primate cells, and some primates will also support viral replication, but it's not primates other than humans are not a natural host. But in any event, so if you want to block infection, one of the best ways to do that is for the antibodies to bind to the virus first and then prevent the virus from binding to this receptor on the cell surface, okay? So that's where the antibody works. Basically, most of the time is preventing the virus from ever getting into the cell. The second place that some of these antibodies could potentially work is after the virus is in the cell, and then to get the replication cycle going, the RNA that's inside of the virus has to be released into the cell. And so if that process is blocked by an antibody that's bound to the virus, that would also prevent the infection cycle from starting, okay? Now, once it's in, and I'm not going to go through these other steps, you make viral proteins, you replicate the viral RNA, and in the end, these proteins and the new RNA genomes come together to form more virus particles. So this makes a lot of virus. If you look at a virus that's been infected in the electron microscope, it's packed full of virus. It just makes a tremendous amount of virus in each cell. It can make so much virus they will come together and form crystals. You can see crystalline arrays of the virus packed inside the cell. And it's so much virus, it just basically causes the cell to lyse. So the cell lyses and all this virus then is released from the cell. So it's a very efficient replication mechanism. But when you want to stop it, if you want the vaccine to stop all that from happening, and that's the goal, you want to stop it either when it's binding to the cell or when it's being uncoated to release the RNA. Okay, so this is a view of the virus. You get a crystal structure of the virus so we can tell where every atom is at in the virus particle. And this is a representation of that. So it's actually not, it's like it's a sphere, but it's a sphere with all kinds of, architecture and shape to it. So there are, low spots, high spots on that. And basically the lighter and the different proteins, 3 different proteins are shown here. There's just viral protein 1, viral protein 2, and viral protein 3. And VP1 is blue, VP2 is yellow, VP3 is red. The colors don't mean anything other than just a way to visualize where these proteins are at. And you can see that. So here we have the VP1 and the others, and there's basically different areas here. But the thing I want to point out here are these white spheres on many different parts here, and some other bright colors here, some yellow ones and red ones. These are all on the surface of the virus. And once we were able to get the exact structure of the virus, we were able to figure out where are these antibodies exactly binding to the surface of the virus. So we know the exact location at the atomic level, that is down to individual atoms and molecules and amino acids, exactly where the antibody binds to the surface of the virus. And those are the key antibodies. So antibodies that can neutralize this virus and prevent the infection from starting that I just talked about, they bind at on these sites that are identified that I'm pointing to on the surface of the virus, okay? So in order to make those kind of neutralizing antibodies, you need these antigenic sites. These are the neutralizing, in other words, these are the sites that make neutralizing antibodies. There are other sites on the surface of the virus that can generate antibodies, but if they won't bind to these sites and they won't neutralize the virus. So you can make antibodies, but if they don't bind to the antigenic, the neutralizing sites, they don't do anything to prevent the virus from infecting the cell. So in order to make a vaccine, that's what we need. We need something that looks, in other words, we got to use basically an intact virus particle. You can't, and these are complex structures. They're combinations of more than one of the viral proteins, and they don't take this. So the structure is complicated. There's more than one protein. It's in a very specific three-dimensional shape. And so it's not like some of the other viruses. For example, with SARS virus, you can take one protein, the spike protein, and a certain region of that, and use that as your antigen to make your vaccine. You can't do that with polio. You can't just take one of the capsid proteins and use it as the antigen, because those antigenic sites don't exist with just one protein. You have to have this exact structure to make those antigenic sites. So it makes it harder to make the vaccine for these types of viruses, okay? And this same kind of polio is one type of virus, but there are many other human viruses, which are human enteroviruses that replicate in your intestinal tract, cause different kinds of diseases. They all basically work the same way, but they have different structures. And then the common cold virus is the same kind of virus as well, and it has similar type of structure, similar type of replication system. So they all have their own unique characters. So for polio, there's only 3 different serotypes. It hasn't wandered or changed out of that definition, basically. So that also has been, we don't have to change the vaccine all the time, like we do with influenza or the SARS virus. This is a virus that is able to replicate and change in many ways, but it doesn't change. If it's type 1 polio, that means when you make antibodies against type 1 polio, it neutralizes type 1 polio, but not type 3 polio. And we only have 3 serotypes. So that part of making the vaccines has been pretty straightforward. Okay, so how well did this work with these 2 vaccines? Worked great. So this is a plot that went from the mid-80s, and there were 400,000 cases a year. And then as we used the vaccine and got more and more aggressive about using it, basically the number of cases dropped to a very low level in late 80s. I mean, we were seeing how well these vaccines were working, and so there was a goal that, okay, well, let's just keep using these vaccines and let's just take it down to 0, and then we'll get rid of polio. And the goal was to eradicate polio by the year 2000. So actually, by the year 2000, the number of cases were really quite low, but they weren't 0, okay? And you can see in 2020, there were almost 2,000 cases of paralytic polio from wild-type virus or vaccine-derived virus at that time. So, when you get close, when you have a line approaching 0, it gets closer and closer to 0, but it's really, really hard to get to 0. And so basically we haven't gotten to 0. And with this kind of a virus, if you don't keep vaccinating, it'll come back. So you either have to get to 0 and completely eliminate it, or you're going to have to keep vaccinating, or it would just come back, okay? So the vaccine, by all standards, it worked great, but it's not, it hasn't reduced polio down to 0. And so the question is, how do we make a better vaccine so we can get even closer to that, or totally eradicate it? Okay, so here's some numbers just to show you kind of where we're at right now. So this is a map where paralytic polio cases are still occurring, and these can be, this is map showing where we have wild type poliovirus type 1 and cases of vaccine-derived polioviruses. So the vaccine-derived polio, the cVDPV is circulating vaccine-derived poliovirus. So this means it came from the oral vaccine itself. And if you look on there, the green dots are circulating poliovirus type 2 that came from the oral vaccine. The red dots are the remaining cases, and that's in Pakistan and Afghanistan, where wild type poliovirus type 1 is still circulating. So this was of July 24. This was the latest map of this type that was available. And then, but you can see we've the wild type virus is restricted to a very specific area where the wild type virus is still circulating, and this is in Pakistan and Afghanistan. So this is a tough place to try to vaccinate people to start with. And there's been, repeated ongoing wars and all kinds of problems in that area. Actually difficult for many vaccine workers to get in there to continue to vaccinate people. And actually in some cases, the vaccine workers have been targeted for assassination because they were they were worried that they were some, in fact, in one case, that was used as a cover, for spies. And so there was suspicion about these vaccine workers. Anyway, it's been a tough place to work, and that's where the remaining cases of wild type polio type 1. But you can see now most of the paralytic disease, the green dots are derived from the poliovirus type 2 from the Sabin strain, and the orange dots are type 3 virus. And so basically all the other cases shown on this map are vaccine-derived viruses from the oral polio vaccine. So that's our biggest problem right now, okay? So it's gotten worse actually over time because we have more vaccine-derived virus now than we had in 10 years ago or around 2015, 2016. Because we had we had basically eradicated type 2 poliovirus. And so the idea was, well, it's generating these vaccine-derived paralytic versions of the virus. So let's take the type 2 out of the oral polio vaccine and just use a vaccine that just has type 1 and type 3. And I was in Geneva at a vaccine meeting at WHO headquarters where this was being discussed. Because I was asking them to give me some money for my lab to do some more research related to this. That ended up being a really bad idea. Because once they took the type 2 out of the vaccine, it turns out you needed to keep the population vaccinated against type 2 virus. And there was a short supply of the inactivated vaccine, and especially to deliver to the populations in the countries where it's, in Africa. And then they weren't vaccinating people. The type 2 version was taken out of the oral vaccine, so people weren't getting immunized against type 2 virus. The wild type virus had disappeared, and so they thought, well, some cases of circulating virus, but it's not going to last very long and it'll just disappear. That was the assumption at the time. Well, that wasn't true because the initial virus that was seen, it kept spreading in the people who weren't vaccinated and kept reverting and getting more and more pathogenic, because you always select for the virus that works the best, and you then select for the ones that replicate the best. And so these circulating strains became very efficient at transmission person to person, and they also regained their pathogenicity because they replicated so well. And so now you got basically a wild type version in terms of pathogenicity of the virus that came from the original vaccine when they were trying to get rid of it by taking it out of the vaccine. Okay, so that's a real problem, you know, actually made it worse. And the only thing then was to think about, okay, how can we approach this? So we knew a lot about poliovirus replication because of people like me who studied basic replication and what does it take for the virus to replicate. Not that I contributed the key developments, but there's a whole field of basic scientists out there working, trying to understand how these viruses worked. And so we knew a lot about, you know, what was important for the virus. And so additional mutations were cleverly inserted into different parts of the viral genome that would attenuate the virus so it wouldn't be as pathogenic, but still let it replicate so that it could generate immunity. Okay, the virus needs to replicate so that you can get a good immune response to that virus. Okay, so now that's been done. So we have what's called a novel oral polio vaccine type 2. I don't know why they picked that exact name, but it was novel at the time they did it. But basically, they put a bunch of additional mutations in the virus so it couldn't revert back to pathogenicity nearly as easy. Okay, and so that vaccine has been licensed for use and is now in use trying to combat this problem with these circulating strains of polio. In other words, we have a new oral vaccine that you can give to the population to protect people, and basically you really want to vaccinate the kids and try to get them all vaccinated as much as possible, and so they are protected against type 2. Okay. Now we don't use that in the United States because we basically had gotten rid of all the polio circulating in the United States and paralytic polio by using the inactivated vaccine. And so basically we use just the inactivated vaccine. But in places, and plus this vaccine, as I told you before, is a lot cheaper to do an oral vaccine. You screw up a lot of this virus and that's what you use as a vaccine. So it's a lot cheaper and easier to develop. So this is the new vaccine. And as you remember, this vaccine was in the news just in the fall, right? There was an outbreak of the circulating form of the oral poliovirus type 2 in Gaza. Remember, they went in to vaccinate every child in Gaza right in the middle of the war, and they stopped the war for like 3 or 4 days or a week, so the vaccine workers could all rush in, and they vaccinated hundreds of thousands of people with this new vaccine against the circulating strain of oral polio type 2. So this is what's been licensed now by the WHO. It's licensed and is in use, and it is definitely better. It's not perfect. It still replicates a lot and can mutate a lot and undergo recombination, but it's got a lot more mutations in it. So the chance of this reverting to wild type and causing paralytic disease, I mean, it's been proven in the field. Basically, it's a much, much, much better vaccine than the original vaccine. So that was a real step forward. And then this can help, reduce these number of cases. Okay, so I want to go back to a similar map. It was just a little bit older, okay? And so this was data as of October 25th, 2022. And what I want to point out is go over here to the United States, and you see there's one green dot here, okay? See it up here where I'm pointing to? That's in New York State. There was a case of paralytic polio in the United States in 2022, and we hadn't had any cases for I don't know how long, a long, long time. We had polio-free, no paralytic disease, and here we have one just a couple years ago in New York. So they tracked, this person lived in a community, religious community that didn't believe in vaccination. And so they had a group of people that were unvaccinated. And this person was one of the unlucky ones. Remember, I told you it doesn't paralyze everybody you get infected, but about 1 in 100 or 1 in 200 people actually get paralytic disease. So there was one person that came down with paralytic disease, never been vaccinated against polio, lived in the United States, and you notice that's a green dot, okay? That's the circulating vaccine-derived virus. It was imported into the United States, into a community where the people weren't getting vaccinated. Okay, so and then they checked the sewage, okay? Because remember I said it's fecal-oral route, and the virus is very stable. So you can just go take sewage samples and see if anybody's got polio in that community. They checked all around the state of New York, and they found lots of virus spread around the state. That meant a whole bunch of other people had been infected with polio, they just didn't have any disease. Okay, so the virus, once imported, it spread through the community of unvaccinated people. So we're living at risk as well, okay? And the lower the vaccine rates get, the more everybody's in danger, and in particular kids that have never been vaccinated. And it's even worse the older you get. So if you're an older child or a young adult and you get polio, it's more likely to cause significant disease. Remember FDR? He got it when he was I think he was I don't know how old he was, 20 something, I think. Anyway, he was pretty young. And he was paralyzed when he got polio. So it's worse the older you get. So anyway, they went in and vaccinated a bunch of people, and they got that under control, but it was the US where we did this. And so we didn't use this new novel OPV, because in the United States we only use IPV, because basically it's safer and we can administer it efficiently. So they went in and tried to get the vaccine rates up with everybody that would agree to get vaccinated. Okay, so how are we ever going to get rid of this thing or get it down to the level where nobody has polio, paralytic polio now? Because a lot of the scientists and people who've worked in this field a long time think it may be very, very difficult to get rid of all the virus on a worldwide level, because so much of it circulates on the subclinical level where it's basically silent transmission. But if we could get everybody vaccinated with good vaccines that aren't causing any cases of paralytic disease, just keep the vaccine rates up high, and in principle, we could, if we do that, we could stop polio in terms of a paralytic disease. So, but basically there, 3 features are basically required if you're talking about eradicating a viral disease. You need safe and effective vaccines. We have some problems with the current OPV, the old original OPV and IPV, as I've mentioned, the problems with that. You need to be where humans are the only host, no animal reservoir, because if the virus can go replicate in a bunch of different animals and then come back into the human population, you're never going to get rid of that virus by vaccination. And you need a vaccine that produces lifelong immunity, and polio does that. The polio vaccines do that. So you don't have to keep vaccinating every year like we do with influenza, and it looks like what we're going to have to do with COVID vaccines, okay? Because the virus is changing so much. Remember, we only have 3 serotypes. It doesn't seem to escape those 3 serotypes. So once you get vaccinated, you should, if your immune system's working well, you should be good. So what we want to do is develop a new non-infectious vaccine to replace the OPV and IPV. and use this for routine immunization. Just use something that doesn't contain any RNA in it, but has that structure. So basically use poliovirus empty capsids, and these are in the field called virus-like particles because they look like a virus, but they don't have any RNA in them. They're empty, so-called. No, they never packaged any RNA, and use that to replace both of these vaccines. And I said, and here on this point, the VLPs do not contain any viral RNA and therefore non-infectious by definition. So they're safe to distribute, they're safe to make because you're never making any infectious virus. But basically the polio VLPs must have the native antigenic structure and be stable at physiological temperatures. Basically, that slide I showed you the colored picture of the virus with the different proteins colored, the red, the blue, and the yellow. You need a capsid that looks exactly like that to use as your antigen, but you got to make it without any RNA in it, okay? And it's got to be stable. So that's the challenge in terms of doing that. And when you make these, then it's going to have to be comparable to the IPV for immunogenicity, because what, that's basically what we want to do is get rid of that. So how do we do that? Okay, I don't want, I'm just going to try to show you one little thing here. So basically when the virus makes its proteins, it makes one long protein to start with, because remember I said the RNA is like a piece of messenger RNA. That's a type of RNA that you can go through a process and make protein from. And so you need this polyprotein, and part of that codes for this capsid, the capsid proteins, and then there are two enzymes or two, it's like, think about scissors that can cut the protein at specific sites. So they're called proteases. That means they cleave proteins as 2A and 3CD. So we need this pink one and this purple protein over here, and we don't need anything else over here, which is the replication proteins. And we need this protein, P1. So we need all of this, and it gets cleaved into three proteins, 0, 3, VP3, and VP1. And you can see this gets cleaved in two spots here and one spot here. So we need the two proteases here, here, and here to make three cleavage events. And then you got the three proteins that you need to make these empty capsids. So all you have to do is express this protein. So it expresses the capsid precursor protein and the two cleaving enzymes, the two proteases. And then you want to get rid of the rest of those proteins in this light blue color because they're proteins that support replication. You do not want this to support RNA replication, because that's how you get the RNA inside of the capsid. It only happens when the virus is replicating. It replicates the RNA, and at the same time it makes a new RNA, then it gets packaged. So you don't want the RNA to replicate. Okay, so that's all you need to know there. And we had a way of doing this. In my lab, we undertook this approach where we have a piece of DNA that has, this is a so-called plasmid. It comes from a bacteria. And we basically are able to insert the sequence of the viral genome convert it to DNA, and then put it into this plasmid DNA. So it basically has the sequence that we want. We can manipulate this because it's DNA, and we can take things out, put things in, make mutations, and then we can convert that, copy that into a single piece of RNA, which is messenger RNA or positive strand RNA. And then we just take that and we can put it either into cells in culture, or we can just break these cells open and have a cell extract and put the RNA in. It still works because the virus normally replicates independent of the nucleus anyway, and all the machinery you need for protein synthesis is in that extract. And then we can add as much of this RNA as we want to make the capsid proteins. So we did that. We set that up in the lab. I got money to start that project. And the logic is shown here because there's a second problem If you have an empty capsid, we knew for years that you could make empty capsids. They just would be made during the normal process of replication. You'd see some that were empty, and most of them were full, but you'd have some empties. But without that RNA, the RNA acts like glue inside of the virus because the RNA and the protein, they can bind to each other. So you have sites on the RNA and sites on the protein where those two will bind to each other. And so that stabilizes the capsid to have the RNA in the inside. If you don't have the RNA on the inside, it's not as stable. So this cartoon, basically what I'm showing here is that we can start with the capsid precursor, the 3 proteins, as one protein, and then we have our protease, 2 proteases, well, one of our proteases here, and we cleave it at 2 sites. So now we have 3 proteins, and then these 3 proteins come together in an organized fashion to form the capsid. Okay, so this would be a wild type capsid because we haven't done anything to change this. And this is illustrated over on the side. So basically the first protein, it folds up into its structure to form what we call a protomer. And then that gets cleaved, and then those come together to form, 5 of those come together to form a pentamer. And those pentamers, then they aggregate with each other to form the empty capsid. So that's an ADS particle, but there's no RNA in there. Now, during normal viral replication, it's making RNA, and you can get RNA into this inside of the capsid when it's coming together. And so you get an empty shell. In other words, you get the RNA package. But if you don't let the RNA replicate, you stop right here, and you basically never get this what's called a provirion. So everything's blocked off, and so you don't get this, and this is the intact final virus. And you stop here at the ADS empty capsid. That's what we want. So stopping replication is critical to get the empty capsids. So the problem was, if you heat this empty capsid that we made over here with wild type proteins, it initially forms in the right conformation. But if you heat it to 37 degrees, which is our body temperature, it goes into it changes because it's not stable. It expands into a slightly bigger particle. And all those little those white dots on the surface, they all disappear. All the antigenic sites that are required to make the neutralizing antibodies, because it goes through a conformational change, you can't use it. It's worthless. It doesn't make any it makes antibodies, but they're not neutralizing. Okay? That's why that's why it was important for you to see that, because you can make empty capsids this way, but they're not stable without the RNA. So the idea was that in other words, this was part of our original application. We had ideas about how to make mutations in the proteins so they would actually stick together better. Instead of having the RNA to hold it together, we changed the proteins a little bit so they would, the individual capsid proteins would stick tighter together and stabilize that so it couldn't change when it got up to 37 degrees. And it worked. Well, one of the mutations we originally designed in our grant application actually worked great. Another one we thought was even better, didn't work at all, and it actually wouldn't form anything. I mean, the capsid didn't come together at all. It blocked the capsid formation. So it worked, and we got an empty capsid that, so basically on the right here is what happened when we had the mutant form, and we got a mutant BLP form. It was stable at 37 degrees, and actually it's stable at temperatures higher than that. And so this figure just shows that this is just the diagram of the RNA, the mutant RNA that we made to be translated. We took out a lot of the replication proteins. We demonstrated in an experiment that we made the proteins that we had to have, the BP0, the BP1, and the BP3 were all there. And we had the 3CD protease and the 2A protease. So we had all the ingredients that we needed. And then we ran the reaction with this RNA and we made empty capsids. And you can see them as these little circles here in the electron micrograph, okay? So basically, we were able to make empty capsids, and we did this at lower temperatures at first, and showed that we could make it with a wild type, and then we did it with the mutants that we were testing, and we could also make it with the mutant forms and get these empty capsids. Now this just shows that it worked in terms of stabilizing it. So this was a key experiment. And basically this is measuring the ability of an antibody to bind, which is a neutralizing antibody, to bind to these empty capsids. And it's called an ELISA assay, but just, and with the output is how many units we have in antigenic units. That means it's in the right conformation. And so the blue line was wild type VLPs. And you can see that after a very short time, all of the inantigenic aspect, it lost all of its inantigenity. It went to 0 in a very short time and stayed there. So that meant it was just like expected. The wild type was unstable at the 37 degrees, which is our body temperature. So if you're going to have a vaccine, it's got to be stable at body temperature. And if it changes to a form where it's not antigenic anymore, then it's not going to work. And it was also stable at 39 degrees centigrade. So this was basically our mutant was in the red line, and then the green line was what's called, it's basically the IPV. It's the inactivated poliovirus vaccine. That was the intact virus that's used actually in the vaccine. So it was as stable as the IPV viral vaccine itself. So that was the key to show, and then we did it at 39 and got the same results, and we did many more experiments like this showing it was stable for long periods of time as well. So we had a stable form of the VLP. So for this, we got a patent for the University of Florida, okay? Because we had given proof of principle, we could make an empty capsid with no RNA that was inantigenic and stable at physiological temperatures, okay? Now, we never thought of the methodology we used to make this as being one that would be a high production type of mechanism for making, but we did it for proof of principle. So we submitted this for a patent, and we got it. It took years to get this approved, but this, in other words, a non-infectious VLP vaccine for global eradication of polio. And this was just taken off the UF website where they have all of our intellectual property, not ours personally, mine, but I mean the university's intellectual property. So basically we got it, and we got the patent approved for that. Okay. Do I still have 5 minutes?
For this slide, yeah.
Okay, this is still a progress and work, and we have collaborators that were working on this to scale up the production. We have people that are using high production protein synthesis, and the best system so far is looking like yeast, just to produce large quantities of these mutant VLPs. And our collaborators, other mutants have been discovered and incorporated into this, but this is the basic idea of how to make them. And so there's a lot of interest from the WHO and now companies that this could be a replacement for the VLP, for the IPV vaccine. The question is, okay, we can go to all this trouble to make the world's greatest vaccine, but if we can't get people to take the vaccine, then we got problems, okay? So this is, and there's a lot of talk about this right now because of the measles cases in the United States, but this is the decrease in childhood vaccination rates in the United States, and you can see that we're on a steady drop. And so part of this was due to the pandemic, and so it really got started during the pandemic because everybody was staying at home and nobody was going anywhere or going to the doctor or whatever. So, you know, the initial part of this was due to the pandemic, but this has continued, and there's, as you know from reading the news, there's a lot of, you know, vaccine hesitancy, or people who don't believe in vaccines or believe they cause, you know, other problems. Even the ones that have been around for all this time, like measles and polio. And so there is real concern, like right now, and this was in the, I guess yesterday, this is in the news right now, the outbreak with measles in the United States. I mean, it's because of this low vaccination rate. The vaccination rate has dropped to the point now where measles virus, we're having, you know, a fairly large number of cases of measles that we haven't had for a long time. So far we've already had 1,000 cases in 25, you know, right now for 2025, it marks 180% increase over what we had the previous year, okay? So basically the vaccine rate has already dropped enough, and there's a paper that's just been published in in JAMA, Journal of American Medical Association. So this is a modeling, but this is for measles. Measles is the most infectious childhood virus there is. And so if somebody in here, if we were all unvaccinated and somebody here in the room had measles, it's likely there'd be a big fraction of us that walked out being infected, okay? And so measles is very infectious, spread by by respiratory transmission. So it's, and you can see here's the current, you know, average rate in the United States. So this is a number of, in other words, what they would predict in this model for how many cases of measles we would have over a 25-year period. And so these are large numbers, as you can see, because these are logarithmic. And so this is 10 to the 6 would be 1 million cases. And if we increase and this is the current levels of vaccination, so if this would go down if we increase the vaccination rate, but it could go back up. And at this level, measles gets to the point where it's endemic. That means it's just there, it's circulating all the time. It would be maintaining a, you know, year over year transmission, and hasn't been endemic in the United States in a very long time. And this is just because of the low current levels. And the other thing to keep in mind, this is an average, so it varies from state to state what the vaccination rates are, and community to community, depending on the attitudes about vaccination. And here is the predicted model for polio. So polio, basically we're controlling it right now. It's not endemic. We're not really having just very sporadic cases, but it is predicted by the model. It takes longer, and we'd have to have a bigger drop in the vaccination rate. But if there's active, you know, campaigning, if you will, or a lot of information being put out saying that vaccines are dangerous and you shouldn't vaccinate your kids, in other words, the vaccine rates could drop. And if we have even a 20%, 25% drop, the number of cases goes up quite dramatically, and then it goes up even further. And so by having something like we would be back to having endemic polio year after year. And so these are kind of scary times in terms of thinking about that. And so, you know, we can have the best vaccines, but if people don't use them, then we could have problems as well. And the other thing I want to say about measles is, and the physicians in the room will know this, of course, but measles is really a very dangerous disease. It's not just the rash shown on that kid. And when I grew up, everybody had measles because we didn't have a vaccine. And we thought about, oh, we'll get to stay home for a week. We'll have a rash, a little fever, we'll feel bad for a few days, and then we go back to school. But that's not why we vaccinated so aggressively against measles, because measles has very serious complications. So a lot of people, and we already had in this case this year, 2 kids died. And so if you don't die, but you have it gets into your brain, you can, you lose your hearing, you can be very severely mentally retarded for the rest of your life. So they're very severe complications. It's not just the rash. And it's important for people to understand that this is a very dangerous disease, just like polio. Polio is a lifetime, if you get paralytic polio, you don't, most people, if it's a serious case, you're not going to regain all of your functions. And so it's a lifetime issue, and just like for kids with measles. So that's why we have these childhood vaccinations. And I think, we have a solution that we've been using for a long time, and we just have to keep basically doing that. Even with the old polio vaccines, we had gotten to the point where this just wasn't a problem anymore. So anyway, on that note, I will end and answer any questions if we have time. And this is just an acknowledgement page with our collaborators. So thank you very much.
Thank you very much, Bert. That was terrific and very enlightening, educational, and I won't go so far as to say depressing, because I think we're going to get through this in a good way, ultimately. So I'd like to take questions.
Hi.
Hi.
I got the serum, we drank the polio vaccine, I guess.
Oral vaccine.
Right. I thought it was interesting because back then, like your parent, they just told us all line up, and we went in the gym and we drank it. There was no permission slip or
Right.
or anything like that, which I was a teacher, and of course things changed. Obviously, they didn't even do shots or anything like that in schools anymore. But when I first started teaching, kids had to have certain vaccines or shots. And of course, as it's evolved, people can have a religious exemption,
Mm-hmm.
which again, when I first started teaching, you either had it or you didn't come to school. So, as you know, a lot of this is parent driven. I mean, if they listen to, vaccines are bad, but it all goes back to the parents not parenting and not doing the right decisions. So I don't know what the solution to that is unless they start saying you can't come to school anymore. But of course, it's usually the child that didn't get it. It's not the other kids. So I don't know. Of course, we teachers always blame parents for everything. So.
Well, so one reason I try to bring this up, whenever I give a talk, is that just to educate people, because I think people think, well, they don't even. I mean, a lot of people I talk to think, well, polio's been wiped out and it doesn't even exist anymore. But there's still polio circulating in other parts of the world, and we just had demonstrated can be imported back into the U.S. The vaccine rates keep going down. We'll have more cases like that. And measles, the other point that I just made, is a dangerous virus, and there are a lot of people that have lifelong, either get killed, either die from it, or have very serious disabilities, from measles. And it's worse in underdeveloped countries, because children that are malnourished and have other, health problems, they're, much worse off. So yeah, just try to spread the word that, getting these childhood vaccinations are really important.
I'm trying, oh, there we go. So there's a question in the chat box. What might be the impact of the end of US funding for Gavi, which buys vaccines for developing countries?
So I don't have any direct personal insight into that, but I think it's just common sense that it costs money to deliver vaccines, and these childhood vaccines are the measles, both measles and polio, and others are part. It costs money to do this, and so if the country you're delivering the vaccine to can't afford it, or the World Health Organization doesn't have enough money to do it, I mean, the lack of funding is going to reduce the level of vaccinations that are, taking place in other parts of the world. And I tried to incorporate that into the talk. It comes back at us, I mean, it can come back. That's where people who are, especially with polio, a lot of times it's a silent infection. And so, in fact, most of the time, so somebody can travel, get infected, the infection goes on for a couple of weeks, you get back and you spread it to somebody else. And then it can spread here in an unvaccinated population. So this is very important to our health as well, to support, these vaccination programs on a worldwide basis.
Okay, well,
Okay,
I think we just want to say thank you very much. And it was a great presentation.